I know we've been here many times before. Wanted to give everyone a very meaningful update today on what's happened since we spoke last. What does Allegro offer? We bring a first-in-class anti-integrin peptide into the retina space. There are no other anti-integrin molecules in retina right now that we know of. We also announced this morning that we have our top-line data in our Phase II DME study in monotherapy. We released those results this morning, and this will be the first public presentation of that data. In addition to the DME Phase II data, which we're showing today, we have also other multiple Phase II studies ongoing with DME in other treatment regimens besides monotherapy. We have a PVD study looking at the progression of diabetic retinopathy as well that's ongoing, and our VMT studies met their primary endpoint and is ready for Phase III as well. To date, across all of our clinical programs, we've treated about 400 clinical subjects in about 1,000 intravitreal injections across all indications so far, and a very nicely tolerated safety profile. Most of our programs are also significantly de-risked from a non-clinical standpoint, meaning we have composition of matter IP, solid PK, tox, and CMC programs as well. Our Luminate program, our Luminate drug has two mechanisms of action, as we mentioned before, anti-angiogenesis and vitreolysis. We're really going to be focusing on anti-angiogenesis today. Before we get into the details of the Phase II DME data, I'll remind you that we also have Phase I DME and proof of concept wet AMD data in humans that also looks very nice as well with monotherapy efficacy. With the data that we've generated before and the data we'll show you here today, we'll show you that we have increased durability, an excellent safety profile, and a completely unique mechanism of action that gives you lots of treatment choices and treatment options going forward. For our top-line data for our DME Phase II study, this study locked about 4 weeks ago. This was a 136-patient study in 32 sites in the US, looking at 1, 2, and 3 milligrams of Luminate, with only loading of Luminate and following at 4, 8, 12, 16 weeks off treatment in monotherapy, looking at vision and anatomy to see how long the drug would last, how the drug would perform in vision and anatomy, compared to the monotherapy group of Avastin every month. This is a monotherapy study, so the Luminate arm of the study does not have any anti-VEGF loaded beforehand, no steroids loaded beforehand. It's Luminate all by itself compared to Avastin. What you'll see here is the study met its primary endpoint. At the top, you'll see in the light blue, you can see that the Avastin arm is getting injections every month, and you're getting about a 6.5 letter visual acuity improvement in the Avastin arm as you'd expect. At the primary endpoint in yellow here, the Avastin arm gives you a 7-letter visual acuity improvement. You really hit your groove with the Luminate arm of about 5 letters, 5.2 letters at the primary endpoint. And there's an interesting thing to point out here. You only load with 3 intravitreal injections of Luminate, and yet the drug just keeps going. At 8 weeks off treatment and 12 weeks off treatment is when you hit your groove. Anatomically, you see the same thing. Secondary endpoint was also met with OCT. In the blue, you can see that the Avastin arm, you get about 100 micron reduction in central retinal fluid, where it basically stabilizes. You can see with Luminate, you give 3 monthly intravitreal injections, and at 4, 8, 12, and even 16 weeks out, it just keeps going by itself with nothing preloaded, no steroids, no anti-VEGF in the Luminate arms, and they again overlap. You can see that at the primary endpoint, there's only a 27 micron difference between groups. At 16 weeks off the last Luminate dosing, you only have an 11 micron difference between groups. Some examples of patients. Some representative examples. You can see that some of the patients were quite sick in this study. The average visual acuity in the study was in the 2040 to 2050 range. The average OCT was up in the high 400s, 480, 490 microns at baseline. You can see a patient here who's on, again, Luminate monotherapy. 8 weeks post the last loading dose of Luminate, the central macular thickness is decreased in roughly half. By 12 weeks off the last loading dose of Luminate, the anatomy is just about back to normal in a pretty severe case. Here's another patient here, different site. It may be a little hard to read the numbers, but again, you start at 365. By 8 weeks post the last loading dose of Luminate, you cut the central macular thickness in half. 12 weeks post-loading, your anatomy is just about back to normal. Here's another patient here as well. I mean, you can just look at the right-hand column. You can see at 8 weeks post-loading, anatomy is coming back to normal, 12 weeks post-loading in monotherapy with nothing else on board except Luminate, and no Luminate for 12 weeks, the anatomy comes back to normal again. So it's nice to see the vision and the OCTs corroborating pretty nicely in monotherapy. In summary, our Phase II DME study with 136 patients met its primary and secondary endpoints of best-corrected visual acuity with less than a two-letter difference between groups and less than a 30-micron difference between groups as well. Also, what ended up happening is we used half as many injections as we did with the Avastin arm. We showed monotherapy efficacy, which is what was the main goal of the study, and 12-week durability. What does all this mean? It means that for the first time, we have a completely new class of drug that has nothing to do with the signaling pathway, but the construction pathway instead. This is an anti-integrin drug. It's the only anti-integrin drug that I'm aware of in the retinal space now, and it's potent enough to stand on its own in monotherapy, giving you nice improvements in visual acuity and retinal anatomy with 12-week and maybe even 16-week durability. We are investigating as well going forward what this looks like in combo and adjunctive therapy, but we really wanted to prove the point here that the drug is potent enough to last by itself and work by itself as well. Thank you.